Well, hello and welcome to everyone joining us for this X Talks webinar. Today's talk is entitled Expanding CAR-T Beyond Oncology, Medical, Operational, and Practical Considerations. My name is Corey Stanton, and I'll be your X Talks host for this event. Today's presentation runs for approximately 60 minutes, and the webinar includes an interactive Q&A session at the end. Make the most of this session by submitting questions and comments for our speakers throughout the presentation, and we'll try to attend to as many as possible. To participate, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Worldwide Clinical Trials, who developed the content for this presentation. Worldwide Clinical Trials is a prominent global contract research organization, or CRO, collaborating with biotech and pharmaceutical companies to advance new medications from discovery to reality. Their full service capabilities include bioanalytical lab services, phase 1 through 4 trials, and real-world evidence studies driven by a skilled team of clinicians and researchers. With over 3,000 professionals across 60+ countries, Worldwide emphasizes a personalized approach to meet client needs and improve patient lives through innovative therapies. And now let's take a moment to meet our panelists for today's presentation. One of our speakers is Dr. Simran Padam. Dr. Padam is the CAR-T medical advisor at Worldwide Clinical Trials, and she has extensive experience in global phase 1 through 3 clinical research. She provides comprehensive medical oversight and collaborative leadership in clinical research with expertise in medical affairs, product development, and strategic planning. She's an active member of the American Society of Clinical Oncology, European Society for Medical Oncology, and the American Society of Hematology. She has authored 15 peer-reviewed publications and has presented at numerous conferences and lectures. And also joining us today is Dr. Amy Raymond. Dr. Raymond has been a drug discovery and development professional for over 25 years, including progressive roles in clinical operations and clinical strategy, and today leads the Cell and Gene Therapy Hub at Worldwide. Her clinical development experience is focused on rare and complex diseases, especially development of cell and gene therapies. Amy completed her doctoral training in molecular biology at University of California, San Diego, and postdoctoral training in molecular biology at Memorial Sloan Kettering. And also here from Worldwide today is Nathan Chadwick. With more than a decade of dedicated experience in clinical research, Nathan is well-versed in several therapeutic areas with a specialized focus in neurology and rare disease. At Worldwide, Nathan is at the forefront of therapeutic strategy for rare disease, leading ongoing efforts with rare disease patient advocacy organizations while working to expand rare disease opportunities, studies, and initiatives. I'm also pleased to welcome Chris Jenkins to the panel as well. Chris is the founder and chief scientific officer at Sabai Global, overseeing subsidiaries like Clinical Biosafety Services, Castle IRB, Alpha IRB, and Shield Consulting. Since founding Sabai in 2017, Chris has grown the organization from a single-person operation to a prominent ethical and biosafety leader in gene, cell, oncology, and rare disease treatments. He emphasizes innovations in AI, systems, and ethical review, aiming to address future research needs. And finally, rounding out the panel is Dr. Tahseen Mozaffar. Dr. Mozaffar is a professor of neurology, orthopedic surgery, and pathology and laboratory medicine at University of California, Irvine, UCI. He is the director of the UC Irvine MDA ALS and Neuromuscular Center and the director of the Division of Neuromuscular Diseases. Dr. Mozaffar serves as chair of one of the biomedical committees and the institutional liaison for Trials Innovation Hub for the Center for Translational Sciences Award at UCI. He is also the site PI and the senior author for the paper for the first CAR-T cell therapy trial in myasthenia gravis and study PI for the Kaplan-Meier-myositis CAR-T trial. And now, without any further ado, I'd like to hand the mic over to our first speaker today, Amy Raymond. Amy, you can kick off the presentation. The floor is yours.
Thank you so much, Corey. And hi, everyone. As Corey says, I'm Amy Raymond. Thank you all so much for joining us today for what I know will be a great cross-enterprise discussion amongst collaborators from a variety of stakeholder positions. All of us here today work on multiple forms of cell and gene therapy, which is such an exciting space. Our conversation today, however, will focus specifically on CAR-T and what we've been seeing as we bring CAR-T to new patient communities. Our hope is that everyone with us takes away insights or perspectives that can be applied to your next CAR-T program. But before diving into the deep end with our experts, I wanted to pause here on the front end, provide a little bit of context, especially for attendees who may be new to the CAR-T arena or even just new to bringing CAR-T to oncology, outside of oncology. Today, there are multiple CAR-T products available commercially in multiple regulatory regions, including the 6 that are available today in the US. these foundational CAR T products proved remarkably effective at eliminating B-cell lineage bad actors. And this is reflected in the range of indications for which these products are approved, things like DLBCL, multiple myeloma, and others. But luckily, innovation continues. And it really is an exciting time as we move into the 4th and 5th generation CAR T designs. While we're focusing today on CAR T beyond oncology, I just want to acknowledge this being just one of many approaches being taken, including CAR modifications for other immune cells, gamma delta, natural killer, macrophage, in addition to many other adoptive cell therapy approaches. The field is seeing a continued rise in allogeneic products. They now represent more than half of the phase 1 trials that are starting, which, said another way, still means that the majority of active trials are still with autologous products, but we anticipate allogeneic products will be what solves some of the current barriers to uptake once approved and help maximize the commercial potential for these products. So looking back over the last 10 years, in the lower blue panel there, you can see a clear growth in the number of CAR T trials started across the industry, plateauing the last couple of years. This year, however, we are seeing that growth curve pick back up. At the end of Q3 of 2024, we'd already seen more CAR T studies started across the industry than all of last year put together. This puts us on track to hit a record number of CAR T trials in 2024. Looking at the panel above, so all trial starts across the industry, that growth in CAR T is not strictly a reflection of the overall growth of the clinical development space, which on the whole is projected to hit the same number of trial starts as we saw last year. This projected near doubling of CAR T trial starts in 2024 is driven more by a combination of the continued innovations in the technology, coupled with increased understanding of how it can be applied to additional patient communities. If we break down the therapeutic areas being addressed by CAR T trials each year, as shown in this distribution bar chart, we certainly see the majority of trials targeting oncology indications, shown there in dark blue. And we are all really excited about what CAR T is doing with solid tumors. But we're also seeing a significant and continued growth in the number of trials being launched in autoimmune diseases. And these span several therapeutic areas depending on the classification system being used. There are loads of implications from a trial design perspective for which indication in specific is being treated by CAR T. And so that's things like the potential need for expert commission functional assessment for many of the indications rightly thought of as being neuroimmunology, such as trials enrolling myasthenia gravis patients. But at a higher level, the challenges of operationalizing these CAR T trials outside of oncology are really quite similar. And that's what the panel is going to unpack today together. To touch briefly on the design of these CAR T products, obviously, different sponsors have different technology details unique to their approach, but by and large, the CARs that we're seeing, these programs outside of oncology are really architecturally similar to what's come before. They're really building on the successes seen in oncology. And as you can see in the pictogram at the top, showing the intracellular domains in gray and salmon, with the antigen binding domain, the extracellular domain represented there in gold. Schett and co-workers surfaced representative examples of that much larger pool of CAR T constructs diagrammed below. Oops, sorry. So we were looking at trial starts per year just a couple of moments ago together. And it's clear that there are now many, many more sponsors in this space. And it's a mixture of those small handful of biotech teams who really pioneered CAR T in the immune-mediated inflammatory diseases. Those are the teams like Cartesian, Cataletta, Kyverna, in addition to emerging and clinical stage biotech teams, and even, as you see there, top 20 global pharma companies pivoting these assets into autoimmune diseases. So those, as a whole, are a really heterogeneous group of conditions, spanning, as I said, multiple therapeutic areas, sometimes seen by very different specialty clinics, right? Nephrology versus rheumatology versus maybe a musculoskeletal clinic. But in all cases with these autoimmune diseases, there's autoreactive B-cell clones and autoantibodies against the patient's own antigen that are driving the disease. So many of the same factors that inspired antibody-based B-cell depletion as a treatment strategy for autoimmune diseases also make CAR T, so achieving really deep B-cell depletion, a promising path for developing meaningful treatment for these patients who no doubt would be really grateful for a single administration option. The value proposition, however, for these patients who have chronic, sometimes debilitating diseases is a little different than for the oncology patients who face acute diseases of high mortality. So while we are seeing an uptick in the allogeneic products moving into the space, for now, many of them remain autologous products. So as shown there on the blue panel on the left, these autologous products outside of oncology still entail apheresis, still rely on patient cells being shipped off reliably to the manufacturer, still require that flawless clinical logistics coordination to get the manufactured cellular drug product back to the investigational site for the patient to then undergo lymphodepletion in many cases, receive product infusion, perhaps even remain inpatient during that critical observation window of days to weeks. Bringing CAR-T to these new therapeutic areas in both rare diseases and non-rare diseases has important implications for operational planning that, if not proactively addressed, lead to unnecessary delays, and nobody has time for unnecessary delays. So collectively, this panel, when we're reflecting on our own experiences over the years and drawing on questions and concerns that come up most often when talking with drug developers, we grouped the primary challenges like this. One thing truly unique to moving CAR-T beyond oncology is the degree of a need for cross-departmental collaboration. That certainly has its own impacts on the processes of site selection and activation, and nobody wants those to be rate limiting either, right? In addition to all the kinds of challenges that are really inherent to cellular immunotherapy, things like that need for clinical logistics coordination, but also really a true need to maximize the data value of that study. So these are what we consider to be the key drivers of a successful program, which I would define as bringing forward commercially successful new treatments to the standard of care and patient access to that safe, effective treatment, usually two sides of the very same coin. With that, we can turn it back to conversation. Imran, I wonder if you might be able to jump in and help the audience get a sense for what are the typical adverse events, serious adverse events that we associate with CAR-T in any indication.
Of course, thanks, Amy. So as we know, CAR-T therapy, while it's a powerful treatment for certain types of cancer and conditions, it can still lead to very well-known AEs. And I'll highlight the most significant and most common AEs that we've experienced, starting with cytokine release syndrome. And depending on the level of cytokine production, this can range from flu-like symptoms to life-threatening shock. And to address this, we have drugs like tocilizumab that we use for managing these patients. Now, CRS is often indistinguishable from infection, so that makes it very challenging to manage complex patients that also present with neutropenia. Another toxicity that we see is ICANS, or immune effector cell-associated neurotoxicity syndrome. And here symptoms can really range from mild confusion, disorientation to fatal cerebral edema, and it can include speech difficulties. So here we require steroids to manage these patients. Now, another potential AE that's termed on-target off-tumor toxicity occurs when the CAR T cells attack non-tumor cells that express the target antigen. So this can lead to cytopenias and infections. And with cytopenias, that can cause increased risk of infections, bleeding, and here we require supportive care such as growth factors and transfusions. Now, cytopenias can be seen acutely, or they can even last up to 3 months. And infections, really the causes are multifactorial. You have lymphodepleting chemotherapy, prolonged cytopenia after infusion, or B-cell aplasia. So all these AEs really require close monitoring and a multidisciplinary approach to manage symptoms and really provide supportive care for these patients. I hope that answers your question, Amy.
It does. Thank you so much. And it starts to lay the groundwork for that cross-departmental collaboration that, Nathan, we're always talking about. Maybe you could kick us off with, what the implications of those AEs and SAEs are for medical oversight from an operational perspective.
Yeah, absolutely. Thanks, Imran, for the explanation. So, this is something that we've been talking about as we see this new wave of opportunities come in with CAR-T outside of oncology. And the question really is, how does a neurologist, how does a rheumatologist understand the complexities of this drug and what it brings for patient management? We've got Dr. Mozafar with us today, who we'll bring in a little bit later on in the conversation. He is obviously, in his site at UCI, was one of the first sites to get running in CAR-T outside oncology. However, as we see from the graphic that you presented, Amy, we are at the very, very start of what is this next wave. And what we really need is we need scalability. And right now, there are a couple of sites around the world that do have experience. However, the majority of sites, as we ramp up to looking at those later phase trials where we're looking to get a lot more patients to get drugs to market, we're going to potentially be going to sites who this is the first time that they're running CAR-T outside of the oncology team. And so there are a number of different factors, which I think is really key to success. Number one is actually, what is the site pathway? And then number two is the patient pathway as well. That's a really big piece that we have to remember when we're talking about these opportunities. So let's first talk about the site side of things. So I think there are a number of different aspects to what a perfect site would look like. Obviously, we want a site that maybe already has or is in the process of getting their FACT or their JC accreditation. That's something that I think a lot of drug developers are going to be looking for, especially those developers who are not used to the oncology space. Now, the second thing that we need then physicians who have access to these patients, which are your neurologists or your rheumatologists. And as you mentioned, Amy, we're potentially going to be looking into other specialties as well, as we just see how far this CAR-T product can reach for patients, with autoimmune conditions and related conditions. And so one of the key challenges is, how does a rheumatologist or neurologist oversee these patients to make sure all of the adverse events, such as the CRS and the ICANS, like Simran mentioned, are managed effectively? And so, Dr. Mozafar, I'd be really interested to hear how you do this. As a neurologist at UC Irvine, how do you make sure your patients on your CAR-T trials are kept safe?
So we are a FACT accredited institution, and we have a standard operating procedure that was put in place specifically for these issues. And we work very closely with our bone marrow unit, so the physicians that are part of the bone marrow unit. Not so much for the RNA-based CAR, but for the DNA-based CAR, we have a very standardized procedure. We usually admit them to the bone marrow unit for the first 5 days of their treatment. They are under the care of the bone marrow physicians. As neurologists, we follow along and advise, but we are not the managing physician. After the CAR has been delivered, after 5 days is over, we will then take over the care at that time. And then we follow them very closely and carefully afterwards. The pharmacy always has the tocilizumab in stock and available if you need to do it on that. And we obviously look for the CRS and the ICANS. Again, being a neurologist, it helps to have that sort of point of view as well.
Yeah, no, that's great. And Simran, we've been talking internally about, the management of these patients and how closely that we do need to manage them. So I know you have a lot of experience doing this and setting up regular calls with the investigators. I don't know whether it would be worthwhile for the panelists, for the attendees, excuse me, just to explain how that works in practice.
Yeah, so when we're managing these patients, during the leukapheresis, lymphodepleting IP administration and post-infusion follow-up period, we usually set up daily calls with sites and physicians to monitor these patients, because again, CRS toxicity is seen very commonly post-infusion. We're checking in with sites via email, and we'll also set up regular calls. And this is usually done, especially for oncology studies, within for at least 7 to 14 days. So we're doing that, and sites are sending us labs in real time, either entering it into the EDC or emailing us redacted copies, basically, to review patient labs, and if they need management, they'll reach out to us and in reference to the protocol as well. So we're constantly in touch at all times, especially the post-infusion time for these patients. And I would say it's like a daily interaction with these sites and these investigators. And I think that open communication allows for effective management for the toxicities that we see.
That's great. Thank you, Simran. Thanks, Dr. Mozaffar. Dr. Mozaffar, I'd like to just send another question your way, if that's okay. Simran mentioned the need for leukapheresis, right? It's a key component of the steps we need to take to generate the medicine and then have the patient ready to get the dose. Now, as a neurologist, I'm sure that you don't routinely use an apheresis unit for this purpose. And so, again, can you just explain maybe to some of the attendees of how this works at UC Irvine and any challenges you may have encountered in the past?
So unlike other specialties, neuromuscular uses a lot of apheresis, especially in myasthenia and CIDP and some of the other neuroimmunological conditions as well. So we have an excellent working relationship with our physicians who run the apheresis unit. They are part of pathology. They are not necessarily part of the cancer center. And that's been some of the challenges, at least in the Cartesian trial, that some of the challenges some of the other sites had, especially in Canada, but also in the US, where the apheresis unit was within oncology and the oncologists were not comfortable using it for non-oncological purposes. We've not had any issues with apheresis. We essentially send the patient over to the blood unit. They will assess for the veins and decide whether the veins, peripheral veins, are good enough for the leukapheresis, and then they will schedule the patient for a leukapheresis appointment on that. And once the leukapheresis has happened, that sample will then go to whichever reference lab it's supposed to go to. But I think that's been the easiest part of our job, only because we had such a long-standing relationship with our apheresis unit.
I want to chime in and just say a few words because I'm so impressed with it, right? The Alpha Clinic at your site is a model for how this can be done really well. And frankly, in my opinion, it was the foresight of the California Institute of Regenerative Medicine to spend years planning these sorts of things. So there's several in California. And I'd say as we look around the country and around the world, it's being replicated to varying degrees. It's a little heartbreaking, honestly, how often these sites have to build from scratch without the benefit of the framework you have there. All of the factors that we're talking about, the oversight, the apheresis, really come into play even earlier in the site identification, the site selection, than budgets and contracts. Maybe you could say a word, Dr. Mozaffar, for our colleagues from other sites who struggle with that.
I'm sorry, Amy, can you repeat your question?
For sure. The budgets and contracts implications to working with the apheresis unit and the ways that that does and does not slow down the budgeting that your team goes through with each new sponsor.
Yeah, I know. I mean, again, I think at least in our institution, it's worked out really well, and it's really a fluid mechanism right now. And part of it also came from our new head of the bone marrow unit was very forward-looking, and he really wanted to use either bone marrow transplant or cell therapy in non-oncological purposes. So I think our sort of interest aligned in that sense. And then Dr. Tran, who's our leukapheresis person or apheresis person, has always been very involved with neurology and MS. We have a lot of patients with neuromyelitis optica that they undergo apheresis quite routinely. So they're very used to doing non-oncological space as well. So, and a lot of my refractory myasthenics get chronic plasma exchange. So I think that really helped as well. So when we get a invitation to participate in a trial and there's a feasibility, we obviously reach out to our apheresis unit. We reach out to Dr. Shuria and his team in the bone marrow unit on that. And then we are respectful for each other's boundaries. I mean, so we know that these patients have to be treated in the oncology unit. There's no point in fighting over that because that's the standard policy. And it's in all honesty, that's actually safe for the patient as well, because they have a different perspective than we have. That, but then they also should be open to consult with us for neurological issues. That's not their specialty, that's our specialty. And we had a recent challenge when one of our patients developed neurological symptoms. Actually was a non-convulsive epilepsy that was confusing. So I think these things happen, and you really have to have an experienced team that's looking after these patients.
So you mentioned some great points there, Dr. Mozaffar. Obviously, at UCI, you're in the fortunate position where, you know, to coin a phrase, this is not your first rodeo. You've done this a number of times, and it seems like you have other members of your team who are very collaborative. I think just a point to pick on for the attendees is, you know, these budgets that we are going to see for these CAR-T trials outside of oncology will be a little bit higher than what we have seen across other budgets, because, you know, we are having multiple specialties who are having to be involved. There are also multiple specialties and multiple sign-offs from departments which are going to be needed as well. So it's not just the total cost, but it's also the total time as well, which is a really important factor for sponsors that are looking into this for the first time. And so from an operational perspective, I think early planning is absolutely key, making sure that when you're choosing the sites, you really understand, you know, how this is going to work at the site, how the patient flow is going to go, who is going to be involved, and ultimately who is going to need to review the budget. Some innovative things that we have done in our past are things like budget review calls. So to support sites with that, rather than a budget, you know, getting reviewed by one department, getting sent to the next, setting up a budget review call where you get all the necessary heads and decision makers on with the contracting team from the sponsor or the CRO, and have a call to really make sure that all their needs are met. And that way, not only do you ensure that the site are left happy, but you usually expedite the timelines as well. So that's a key important piece. To go back before we kind of move on from this, I do also want to talk about the patient journey at the site. And I think that's especially important. Again, we heard from you, Dr. Mozaffar, how fortunate you are with your apheresis unit. But there are some sites that are not going to be so fortunate. There are some sites that maybe don't have access to one nearby, or don't have access to one on site. And so I think because the patient population may still be there, it would be remiss of us as drug developers to leave these sites off the table just simply for the fact that they don't have this unit on site. So that piece in the patient pathway, you know, we really again have to understand that from the site level. That involves in-depth conversations between the CRO and between the sponsor to find out where the patient is going to need to be sent, if it's to a separate hospital. We have seen, again, as far as you referenced in Canada, apheresis delays. And so, you know, can we utilize centralized apheresis units? And so here in the UK where I reside, we have the NHS blood transplant team. They have 8 apheresis centers which can be tapped into. Similarly, in America, we have the American Red Cross leukopaks, and again, they can be used as a centralized option for those sites that maybe don't have that ability on site. Or the other challenge, and I think you alluded to this as well, Dr. Mozaffar, is their unit is so busy anyway for clinical trial purposes, these patients may not be anywhere near to the front of the queue, and ultimately we don't want them not getting treatment. So these are the things
if I may add, part of the challenge is a lot of the cancer centers in the US are funded by a P50 grant from the National Cancer Institute. And by definition, it's only supposed to be used for cancer-related purposes. So there is an inherent hesitation to see patients. I mean, we saw that in the infusion centers when we were referring patients for non-oncological treatments. We've seen this with leukapheresis and other things, because their mission is to take care of cancer patients. I mean, this has happened in Boston with Dana-Farber. This happened in a lot of other institutions. And then in terms of budgeting, the first time we did the Cartesian study, we were intending to do it, being naive and everything, we wanted to do it as an outpatient. And then somewhere along the line, there was a mandate that you have to do it, the first treatment at least, as an inpatient. And we had never programmed in the budget things like chest X-ray and some of the additional microbiological testing, etc. That came as a surprise to us. We had to go back to the to the sponsor and do it. Sponsor wasn't happy, but we somehow managed to do it. One advice I would give is that these cell and gene therapy protocols are very involved. There's a lot of coordinator time that is somehow never budgeted or not anticipated and not budgeted. And there's a lot of offline work that even the PI has to do. So at least at our institution, we tend to put in a flat 10% PI oversight fee above and beyond what the individual PI receipts are. And then similarly, the coordinator time has to be carefully watched because you're going to take a loss if you're not careful with your budget.
That's the value of experience. Thank you for sharing that. Yeah, I think that's the sort of thing that, to chime in on a couple of points that Nathan's making, when we're doing those kickoff calls that Nathan references, we're sometimes working with sites that have done it before, such as yours, and sometimes not. And why is that? Because, you know, as Nathan's saying, a lot of programs are looking for FACT accredited sites. Yeah, so that's the accreditation here in North America. I'm old enough to remember when JC, the European, they weren't harmonized yet. Luckily, they're harmonized. That makes things much better. But there are times when it's really just a function of the clinical development program. If they're going to be doing a freestanding phase 1 and know that they're going to have multiple additional trials, it makes a ton of sense that they're not yet focused on bringing their product to where they will do it commercially at the FACT accredited centers. But those kickoff calls, sometimes for the centers that aren't as experienced, we're talking them through. Now, please look internally. Who will need to be the one who decides this value? Because even though it takes a little bit longer on the front end, it shortens the overall timeline and meets everybody else's needs. The fastest path there.
I think that goes into the IRB and IBC approvals too for these two. If you're not FACT accredited or have the experience with these types of trials, you could go extra months getting approval from one or both of those regulatory committees, IRB for the ethics review and informed consent, and the IBC for the genetic modifications in the US, or GMO committee in UK, or wherever your country is being resided, and that'll delays as well.
Once upon a time, I think, and I mean when you founded the company in 2017, we were often turning to you and your team to help sites because they hadn't done it yet. And it's funny to see how it's come full circle from, and can you and your expert team, Chris, help this site because they haven't done it, to now they do so much of it. They're very, very busy. Can you help them do it very well? Now back to, okay, they're very, very busy, and the specialty clinic is one who maybe hasn't done it before. So that's been an amazing evolution.
Yeah, what we're seeing a lot of, like, and this goes a little bit beyond, CAR T oncology and into other gene therapy as well too. But you're getting groups involved that, while the center, oncology cancer center may have experience, or the neuromuscular physicians have experience with drugs like Zolgensma and others from those clinical trials, you're starting to get outside clinics, you're starting to get third party, apheresis or other groups involved, and the contracting for a third or fourth party is mind numbing. Unfortunately, if you're starting to pull those in who haven't done it before, so getting them early, getting them prepared to, hey, this is a little different than what you might be used to is key.
Absolutely. The more we can do to help teams not have to go through what Dr. Mozaffar and his did in that learning curve, I think the better it'll go overall. Certainly we have alliance sites, so part of that larger network, some of whom are FACT accredited, some of whom are in that same situation where it can actually be faster for booking the apheresis that they use a third party, but as you say, the contracting is not trivial. It's one thing that I think we're all looking forward to is the upcoming changes in the IRB and IBC, I want to say, ecosystem in America with the recent guidances. Can you share a few words with the audience about that?
Yeah, so with the upcoming harmonization by the FDA to the NIH, so there will be a single IRB review on multi-center trials irrespective. So rather than having the IRB done at every local site, it'll just be required that there be a single or central or independent IRB that conducts that review, and then each site is brought on to that. On the IBC side, we're seeing a rise in the US as well as in other countries, Canada, UK, Australia, in the utilization of a master protocol review of the study. What is the drug? How is it genetically modified? What's the lentivirus or the vector? How is it tested to make sure that there's no residual virus left over? And then being able to add sites specifics on how are they going to receive, store, prepare, administer that drug. And that's helping to accelerate both the individual site startup and being able to do that, as well as reduce some of the regulatory overhead and burden, as a lot of those costs then are passed through directly through the site to the CRO and to the central provider in many cases. So we're seeing that reduce some of the study startup timelines as these trials go from phase 1 at UC Irvine and a couple other sites to phase 2 and phase 3, where they go to 10, 20. So we've seen some of these studies get up to 50 to 100 locations, and that's really where you see the orders of magnitude of acceleration for starting these trials and getting the enrollment to get the data that we need.
They are complex. Products coming out of them are well worth it, right? It's all time well spent, but everybody's looking to optimize it in any ways that we can. One other factor that can sometimes come up is recruiting the patient. Nathan, you touched on the patient journey. Maybe realign us on that topic.
Yeah, so I mean, this is really interesting, right? We are moving this therapeutic away from people who have life-threatening diseases to patients that have life-limiting, and certainly, the way that their disease manifests stops them doing day-to-day, but it certainly doesn't give them the same prognosis as the foundation on which these drugs were developed. And so really the question that we have to ask ourselves is, why would a patient with myasthenia gravis, why would a patient with lupus or systemic sclerosis want to join a CAR T trial? And like everything, it starts with education. So as mentioned in the preamble, my background comes from rare diseases. And a lot of the time we can apply similar methodologies that we apply in rare disease to this space. So in rare diseases, the big challenge is a lot of the time there is no medication that's out there. And so every medication needs an explanation. I think one thing which we're forgetting, or perhaps forgetting with this medication is while it comes with a wealth of research and background in the oncology space, these patients, and for sometimes the sites and the physicians, are going to be naive to it. And so really explaining the drug, its mechanism of action, why it's now being researched, how it can be utilized alongside their current therapies as well. I think that's really, really big key point for these patients. Obviously, that's going to vary protocol to protocol, but a lot of the times, as long as they're on a stable dose of their DMARD or whatever it is, they're going to be allowed to carry on and join the trial. Some really cool things that we now can start doing is in the digital age of 2024, we can start to use technology to support us. So setting up websites, we can use graphics. Amy, very much like the graphics that you were sharing on your screen, but in a patient-facing manner and via video form to actually explain the process. We are going to take cells out of your body, we're going to ship them off, and when we put them back in, we have given your body the code to fight the underlying cause of your disease. And explaining that in a really nice and easy way for patients, I think, is going to be absolutely key. The second thing that we see in a lot of, not just rare diseases, but a lot of studies where we have really invasive procedures as well, is breaking down those procedures, potentially even doing patient testimonials that can all be put on a website. Of course, Chris, that would have to go through IRB approval, but we'd make sure that step gets done. But again, that's going to give these patients a sense of confidence. And the final thing that I'd like to touch on before I open it up to the rest of the group for input is, obviously, one of the big barriers of not just CAR T outside of oncology, but CAR T in oncology as well. They're very front-loaded. So They're very, very heavy, as Dr. Mosafar was saying, inpatient stays for 5, 6, 7 days. Some, molecules dependent on the generation may require the patient to stay close to the hospital within, an hour of the hospital for 28 days, potentially even longer just afterwards. Now, we cover that really, really well. What we don't cover is the follow-up that's needed and the mandated follow-up that is needed by regulators globally for these products. So the 15-year follow-up is a really, really, really long time. And with the diseases that we're investigating it in, because they don't have the same mortality rate as oncology does, unfortunately, I feel like we're in another unprecedented situation where we are really going to have to follow a good majority of these patients for that full 15 years. And do they really understand what the ramifications are of that? And I'll now open it up. And Chris, I have a question for you, if you don't mind. Informed consent underpins everything that we do in research. And so as an IRB leader, what do you think sponsors really need to be doing with the informed consent, especially if we're talking about one protocol or split protocols?
Right. I think they have to be thinking the full length of the study from the very beginning. Where are they with these patients, especially if they are going 15 years, they have to make that education up front, and they have to write into their protocol how they're going to follow these patients up for that period of time and keep up with them. We just don't want to lose these folks to follow up and lose that data. So you got to think about that early on. I think that there's been some items in the news recently that have been unfortunate for the ethics community or the ethical community, that we need to work collaboratively with sponsors to make sure all those risks are identified in the protocol upfront, and those are outlined. But in these cases, it shouldn't be as major of an issue. But we do need to outline those and have that information in the informed consent.
And Nathan, sorry, and Chris, I just want to kind of piggyback on that. Nathan, you did talk about education, right? So, and then Chris, you talked about informed consent. So I think these conversations on educating these patients should start early, especially during the informed consent. Because if you're not informing the patients about the clinical trial, about the therapy, potential adverse events, is it really an informed consent? So you kind of have to think about that. So I think when patients are educated, they're knowledgeable about what's expected of them, what toxicities are expected, and what type of follow-up is expected from them, they're more likely to be involved in the trial, more likely to follow up on their visits, and stay for that long-term follow-up. As well, they feel more involved. They feel like they're not just a patient number or a participant in a clinical trial. They're actually making a difference. So I think that's something that needs to be communicated, especially during the informed consent process, because that can help with long-term follow-up where we lose a lot of these patients, because the follow-up time period is 15 years or so. So I think these conversations definitely need to start early on during the informed consent process.
Yeah, I have a question for Dr. Mosafar. When you're offering clinical research as a sort of treatment option or a potential pathway, do you find patients come to you first, like, I'm interested in something, do you have a trial for me, or do you find that your team is doing a lot of educating of kind of the options for patients as they come in?
So we've seen all sorts. So most of these are our own patients who have been refractory to medical treatment and doing poorly despite being on good drugs, and we would broach them with the idea, look, here's this new experimental therapy that has a lot of promise. We have a lot of experience with it, or at least in the case of Cartesian, we would say, look, it looks promising, seems relatively safe, try it. A lot of people get concerned about the whole chemotherapy business and especially the preconditioning chemotherapy. And as you mentioned, one of the moves in the CAR-T field is, do you really need both cyclophosphamide and fludarabine as a preconditioning, or can you get away with just cyclophosphamide? And most neuromuscular physicians are comfortable with cyclophosphamide because we use so much of it in the clinical practice as well. But we've also seen patients who have come out of the woodworks and basically said, we are interested in doing CAR-T because they heard about it and they think it's a very attractive treatment. Unfortunately, a lot of those patients that self-refer themselves usually don't meet the criteria for CAR T cell therapy. So again, the last thing you want to do is to fill your trial with patients who are going to screen fail. So we usually have a very good system of seeing them in advance or screening their records in advance and make sure we have the zero amount of screen fail if we can.
It's no trivial thing. That's a lot of work for your team. It's valuable work. It helps maintain patient safety. It uses everyone's time well. I just want to comment that now that we're moving these CAR T products, not just outside of oncology, but sometimes also out of rare diseases, large disease patient populations like multiple sclerosis. It's heartbreaking how large that community is. There are times when we look at it and we say, there are going to be a lot of self-referrals, and in those scenarios, it can be helpful for us to set up a centralized medical, like a nurse navigator, to review the medical records with them and to share that information to the site, just to cut down on how much of that back and forth is happening at the site, but making sure that everything's being handled HIPAA compliant and bringing the true information to the clinic without having to ask the clinic staff to hunt it all down from patients all over the country. So just a little ways we're handling that.
So I think as we get more experience with CAR T cell therapy and as we continue to show that it's safe and relatively harmless, you will start seeing relaxing of the attitudes. Right now, we are primarily using it for refractory disease. But like what we saw with the complement inhibitor therapy, like what we saw with the FcRn inhibitor, rather than taking refractory patient, they will, I mean, FDA is a good example. The Solaris trials or the eculizumab trials were truly meant for refractory myasthenics who were not responding to other treatments. 2 days before or 1 week before approval, FDA told them to remove any word that says refractory. And they essentially became approved drug for generalized myasthenia. So I think we will see something like this in the CAR T field once we get more approval and more experience with this.
And I think that can only be a good thing, right? Because at the end of the day, you know, these patients usually with these autoimmune conditions, once they have it, they're on a path, and it's only leading them to further complications. It's only leading them to more life-limiting problems. And so if we can treat the patients earlier on, I think that's absolutely something that we should all be doing. So I think long term, again, once we know more and once these drugs start to get investigated in these conditions outside of oncology, I think that's really somewhere I'm excited about for more options for these patients.
I also think too that the industry is funding. We're seeing it in the latest ASGCT landscape reports. There's more trials happening. There's more funding happening in pre-seed and Series A rounds. So, you know, not just centers like UC Irvine that have done the rodeo have the fact of credit, but even centers that maybe traditionally haven't thought about this. Maybe non-academic medical institutions or academic medical centers that have historically not. You need to start thinking about how are we going to both treat as an approved product, but also how are we going to run these trials, because eventually we're going to hit a, you know, there's only so much that Dr. Mazzafer or your team can handle, and this is a great situation to have more trials starting to come for these patients, but they need more locations in order to operate.
I couldn't agree more. I think that eventual of saturation, we're kind of there in the next year, honestly. In my opinion, as we move into these additional patient populations, I can say that some of our programs have started incorporating even CAR T experienced community hospital, right? For all the reasons you just said. Great point.
I would say that's probably one of the biggest topic areas is how do we bring, you know, CAR T oncology and non-CAR T oncology to the community centers, you know, where a lot of these patients for geographic or financial reasons can't make it that UC Irvine. Right?
Looking at the clock, I want to make sure there are a couple more topics that we get to for the audience today. One of them being maximizing the value of the data collected, right? The data value of the study, because Chris, you referenced the all-in-one long-term follow-up. It's a master protocol for efficiently rolling participants from multiple studies into one trial, and we're always looking for ways to optimize that way. As you pointed out, right? If we lose the patients along the way, well, we've lost data that informs safety, but also that sponsor has lost data that informs their conversation with the payer, which then in turn affects things like patient access, right? Even if it is effective. What are some things that our team thinks about, Nathan, for how we maximize the value of the data?
Yeah, so I think first and foremost, it's a stepwise approach, and it starts, I think, first by training, making sure that the teams are trained really well. And by the teams, I mean our CRAs who are going out there and looking at this data. And we have a comprehensive strategy, which starts with the 360 training that we provide to our CRAs. And so Simran, I'd like to punt this over to you to talk a little bit about that.
Yeah, so our 360 comprehensive training is training that we perform for our study teams, so internal teams, including project managers, CRAs, and the rest of the study team, basically. So this includes a training on the protocol with the study design and the patient pathway, and so what to expect in this particular clinical trial. We have a training on the therapeutic area that's being studied. And then this training also includes a modality training, which is the actual product, what's the mode of action, what are the potential AEs that you may see with using this product. And basically this comprehensive training provides a very in-depth, I guess, discussion or review of the protocol and of the clinical trial that's being conducted. And then I'll hand it off to Amy to add additional information on our trainings that we have.
Oh, sure. Happy to do. Yeah, because there's training for the study, but then there's planning for this massive wave. Then there's, how do we, even as an organization has really low turnover, how do we plan ahead for when there is going to be transition? Because there is. I mean, there's promotions of people. So how do we keep all of our staffing up to date? One of the ways to do that here is to have foundational training for the right staff members. So this is a multi-course knowledge checked series of trainings that really introduce all of the foundational topics, molecular biology, in addition to the operations, in addition to a review of the technologies, which gives CRAs the chance to then, if selected based on high performance, go shadow and have the lived experience of these studies without it being at risk for anybody, right? It's just part of our investment in continuing to grow the army of people we're going to need as we see that wave continue. Not great news for patients, but getting there efficiently is the trick, right?
Yeah, thanks, Amy. And so I think once we have our teams trained, the next side of it is in setting up is strategy. And to me here, the strategy comes in two forms. It's working as a strategic partner with the sponsor to make sure that they've got the right amount of data cuts in it. For that long follow-up, you really don't want to be waiting 15 years to wait to get to the data to start to publish it. So working with them to understand, what conferences are upcoming, what investment, if needed, are they targeting, and when is that coming? So we can really look at future-proofing, but also planning ahead. The strategy then also comes internally. So as I mentioned before, these CAR-T trials and really a lot of cell and gene therapy trials in general are very front-loaded, especially the one and dones. There's a lot that goes on in the first couple of months just around dosing, and then we go into the long-term follow-up. And so when we approach this from an operational perspective of monitoring, we almost need to make sure that we have this flexible monitoring strategy. So we have the monitoring strategy for the dosing and post follow-up, and then we have the monitoring strategy for the long-term follow-up, so that we're not overburdening the sites, going out, we're not artificially inflating budgets visits by sending teams out on lots of different visits when they're not really needed. And then also what we always would recommend in trials like this is a pool of visits that can be tapped into at any time, because things happen. As we come in, as we start seeing safety data, Chris, you alluded to it before, they may be forced to do a data cut and a DSMB and send things off to the regulators very, very quickly. So having a pool of monitoring visits that as a CRO, we can go out and we can implement almost a study-wide, site-by-site deep review of the data without having to go and change order a client for extra money is really, really key. And then just quickly, because I know we're running tight on time, is following that up, right? So you've got the training, you've got your strategy. Now it's making sure that we really kind of, follow through with what we said we were going to do. As always, we would like to maintain a healthy SDV percentage of above 95%. That's something that means sponsors have ready access to clean, accurate data that they can put out in the public domain. It also means that the safety review committees in their various different forms, they have access to reliable data as well to make their decisions. And so we do that by really going through on a very regular basis and forecasting the visits that are going to be coming in, the SDV backlog, and therefore the triggers that we need to make. I think, and I'm really glad that there has been an industry shift moving away from the classic monitoring of every 10 to 12 weeks. As we see these dynamic programs, as we see adaptive trial designs, as we see basket protocols, we need to move with the times and we need to be flexible. And in part, that is also supported by the mandate of RBQM, and the RBQM systems that we can use, we can now also utilize to support data-driven monitoring at the sites. So those two pieces coupled together really make sure that we have a keen eye on the amount of data that is going to come in. We forecast that, and then we plan appropriately for making sure that we maintain that really nice level of cleanliness.
Wonderful. Thank you, Nathan. And I recognize we're down to our last couple of minutes. So hopefully in the time together, the audience took away some things around the cross-departmental collaboration, ethics, so important, the ways that we think about the patient journey for these trials. But I wanted to turn it to Corey. Corey, have any questions come in from the audience?
Yes, thank you so much, Amy, and thank you everyone for that really excellent, insightful presentation. We do have a couple minutes left here, and I would like to just hit some of the questions that have come in from the audience. To our audience members, though, if we don't have a chance to get to your question now, or if you have any further questions, rest assured you'll have a chance to follow up after the webinar. So I think we'll have time for a couple here. This question first was one that I was actually wondering throughout the presentation. I'll direct this towards Dr. Mozaffar. This audience member was asking about, given your direct conversations with patients being recruited for CAR-T trials, how do you actually introduce this new technology to them in a way that they can understand, in a way that's comprehensible to someone who isn't on behind the scenes like the team here?
Well, I would say it's not easy, but you have to bring it down to what we generally follow in the IRB as a rule, is to eighth grade level, and you have to tell them what's going to happen to the blood that we're going to take out from them, how we're going to engineer it to become effector cells, to recognize autoantigen and destroy them. So, I mean, keep it simplistic, keep it maybe in popular terms. I mean, you can borrow a lot of stuff from smart bombs and video technology, video game technology. The idea here is that you're taking your own cells, you're engineering them to start recognizing these foreign cells or the foreign antigen and selectively target. I think the emphasis is selective targeting. It is not like your regular chemotherapy. It's not going to go after all your T cells and all your B cells. It's only selectively going after certain B cells.
That's great. Thank you. I like the idea of the video game analogy. I think that's really cool, and probably it lands well for patients. Unfortunately, I think this is probably just going to be our last question here, just based on our time. But I think maybe this is a great one to open up to the panel and anyone who'd like to chime in. But Amy, I'll send this one to you first. This audience member was asking if this trend of non-oncology CAR-T therapy, if this trend continues, where do you see this next wave of innovation happening in cell therapy? What's on the horizon here?
Yeah, it's a great question. I definitely did not come equipped with a crystal ball. Often wish I did. I think some of the advancements we've made in the last, let's just say 10 years, are things that just blow my mind, right? In the best of ways, innovations, advancements that I really did not perceive being this close in the future. Some of that I'm referencing, like the Casgevy approved last year, to have sickle cell patients now, in some cases, functionally cured by these treatments, is the sort of thing that really lights the flame under all of us to keep innovating. But I would say the transition increasingly to allogeneic, I would say the transition to multiple cell types. We're now going through adoptive cell transfer types of technology. But I would also, this is maybe a little out of scope for this conversation, more and more in vivo CAR T, where what we're delivering to the patient isn't actually the cellular product, but rather the nucleic acid machinery, so that it can be made inside the patient's own body. I think what we're starting to see there, whether it's in vivo gene editing, but the in vivo CAR T type of products, I think are very exciting for what it could mean for safety. Thanks.
That's great. Thank you, Amy. Before we wrap up, Chris or Nathan or Simran, any thoughts on where we may be headed in the future? Anywhere you wanted to chime in there? Oh, Chris, I think we got you on mute.
I think there's a lot of interest towards, dual or bispecific CARs, targeting multiple antigens. I also think we're looking at, the in vivo CARs, we're looking at allogeneic, off the shelf. So there's a lot of different techniques that I think are still being teased out that have yet to come, and I think are promising in their own ways.
And then I think we're also seeing, oh, I'm sorry, Amy. I was just going to add that also combination treatments that we're seeing a lot more recently. So I think that's going to be the future as well. Sorry, Amy, to cut you off. Go ahead.
No, I was just agreeing with Chris. Bispecifics and even trispecifics. You and I have work that we do together on trispecific CARs. Sorry.
That's great. Thank you all so much. Obviously, a topic that we could talk about for much longer. Unfortunately, though, we have reached the end of our time here for today. As I was mentioning earlier, if we couldn't attend to your questions in the audience, if you're someone who has further questions, or if you're someone who's watching the recording of this webinar, you can absolutely get in touch with the team at Worldwide using the contact info up on screen. We've got the Simran, Amy, and Nathan's info up there. Feel free to reach out, and they'll be happy to follow up with you after the webinar. I do thank everyone once again for participating today. If you are attending, for those of you who are attending here for GoToWebinar, take a look at the chat box where I've added a link. You'll be able to view the recording of this event, and you can of course share this link with your colleagues so that they may register to view it as well. You will also receive a follow-up email from XTalks, and after we wrap things up here, a survey window will appear on your screen. Your participation in the survey is always appreciated, as it helps us to improve our webinars. Now, please join me in thanking our speakers once more for their excellent discussion. Amy, Nathan, Simran, Chris, Dr. Mozaffar, really appreciate your time today. We hope our audience found this insightful and informative, and we wish you all a great rest of your day. Take care, everyone.
Thanks so much.
Thank you.
Everyone, bye-bye.